Hi guys, today we are going to talk about Mendelian genetics. So Mendelian genetics is going to look at how traits are inherited. In this lecture, we'll talk about Mendelian genetics, or sometimes you'll also hear it referred to as complete dominance. And then later on in the unit, we will talk about the exceptions to the rule. So let's go ahead and get started and learn a bit about who Gregor Mendel was and how he really is considered to be the father of genetics. So when we talk about heredity, we're talking about the inheritance of genes from our parents and how our parents inherited those genes from their parents and how those genes or traits go along our family line. So basically that is what Gregor Mendel did, but he did it with pea plants. So he was raised on a farm and understood the value of plant breeding. And so when we talk about that, really what Gregor Mendel did is he took the best plants and bred those together. Okay, so selective breeding. So he was choosing, let's say, the tallest plant with another tallest plant, and he would cross-fertilize them. Or he might actually take a tall plant and self-fertilize or fertilize itself. And so he had an understanding of that in the selective breeding process. At 21, he entered priesthood and studied plant breeding in a monastery in the Czech Republic. So he actually was a monk. And he loved to read, and he was very interested in natural sciences. And he was also aware of Darwin's findings. And later on, we will talk about how Darwin, who's considered to be the father of evolution, is related to Gregor Mendel, who is considered to be the father of genetics. And like I said before, Gregor Mendel studied the inheritance of these traits, things that he could see in pea plants. Now, Gregor Mendel was extremely lucky by choosing the pea plant because he studied the traits of the pea plants and they occurred in two distinct forms. So if you look over here, these are some of the traits that he looked at: plant height, flower color, flower position, pod color, pod shape, seed color, seed shape. And so each trait has two distinct forms, so tall and dwarf, purple, white, axial versus terminal, and so on. You guys get the idea. But just be aware that not all genetics are like this. It's not very black and white. And so he was very lucky in that he chose a plant that had two distinct forms of each trait. So let's talk about some definitions. So in the previous assignment, you guys had to do genetic vocabulary. The reason why I had you guys do that is because the vocabulary is very important in understanding what's going on in genetics. And so also be aware that some of the terms are interchangeable, such as gametes. Gametes, you might also see the term allele. So just be aware that those two are related terms. And so a gamete is a reproductive cell by sexually producing organisms, male and a female. So you're going to have a male gamete and a female gamete. And so the male gamete is sperm, and in plants, it's going to be contained in pollen or pollen grains. And eggs are going to be contained within the ovules. Okay, so if a flower is a complete flower, it has both male and female parts, and it's able to self-fertilize. Fertilization. So fertilization happens when that sperm and egg fuse, and it will become a zygote. I just talked about this a second ago, but self-fertilization means that sperm and egg from the same plant are going to be fertilized. So it's fertilizing its own parts, okay? So it's not going to come from a different genetic source. If we talk about cross-fertilization, that is the fusion of egg and sperm from two different plants. So just be aware that when we talk about different crosses, some are going to be self-crossed, or self-fertilized, and then some of them are going to be cross-fertilized, meaning two different plants are used. Two more terms, you'll see F1 and F2, and these just mean first generation and second generation. And so this is how Mendel actually cross-fertilized. And so what he would do is he would take the male parts off of one flower, and he would take the pollen or the male sperm from this flower and put it on the female part of this flower. And so this is how he cross-fertilized. If it was self-fertilizing, he actually wouldn't have to do anything. But if he did, he might take his little brush and kind of take the pollen and put it on the female part of this flower. So that's exactly what he's doing. And so we talk about his analysis of the offspring and how he put them into mathematical terms or ratios or probability. And so you'll actually see that in the later slides, but here's kind of his observations and how he related them to mathematics. So he is going to take a wrinkled pea and a round pea and he's going to cross-fertilize them. Okay, so these are from two different plant sources. And his F1 generation, or that first generation, have all round. So even though we originally have wrinkled and round, we end up with all round in our first generation. He then takes these seeds and he's going to self-fertilize. So he's going to take one plant from the F1 generation and self-fertilize. And then in that F2 generation, he sees 75% round and 25% wrinkled, or 3 to 1. And he actually saw this outcome many, many times. All right, so what did Mendel interpret from these findings or his observations? And so we kind of have to interpret his findings into our modern day vocabulary. So he wasn't using the term genes or alleles, but now we can use that. So genes are going to be the hereditary information that determines a single trait. Okay, so that DNA is going to be contained within a gene. Alleles are the alternate forms of a gene. So if we're talking about pea plants, let's talk about plant height. Okay, so a gene is going to code for plant height. Well, there's two different alleles, okay, a tall and a dwarf. And so again, Gregor Mendel was extremely lucky because he wasn't dealing with a plant that had many different variations. It was only two. Okay, so two different alleles for that gene. So when an organism inherits two identical alleles for a trait, we can say that that is a homozygous. Okay, and what that means, homo means the same, zygous kind of refers to that zygote, that fertilized egg. And so they contain the same allele. When an organism inherits two different alleles, and there's an error right there, and I'll fix it. So when an organism inherits two different alleles for one trait, the organism is called heterozygous for the trait. So hetero meaning different. Okay, so the alleles are different. So now let's talk about alleles and their dominance. So this is also very important to know, dominant versus recessive. So a dominant allele is going to be expressed whenever it is present. So remember, we inherit half of our information from our mom, half of the information from our dad. Now, it doesn't matter, let's say we inherit that dominant gene from our dad. It doesn't really matter what our mom gives us because that dominant dominant allele is going to mask a recessive allele, let's say if our mom gives it to us. So, and the only way that recessive alleles are going to be expressed is if there's two recessive alleles. So again, dominant will always mask that recessive. Now, another term you're going to see is phenotype. So phenotype is our physical trait. So in this case, his phenotype would be that he's spotted, okay, or has spots. The phenotype of this guy would be that he doesn't have spots. And you also hear the term genotype. Genotypes are made up of the two alleles, and that will become more obvious when we start looking at specific genotypes and phenotypes. Again, so that genetic makeup of an individual is determined. So we have two alleles that make up our genotype. The genotype determines or codes for our phenotype or our physical trait. Okay, so it's the expression of the genotype. So it's that outward appearance that we see. So now let's talk about how we can track genetic probability. Okay, and we do that through something called Punnett squares. And you guys are going to practice in Punnett squares in the next part of the unit. And so this is a Punnett square right here, and it's showing the genotypes. Okay, this is an allele and this is an allele. The two alleles together make a genotype. This is an allele, this is an allele, and the two together make a genotype. Now, if we look at this color, it's yellow, and that's its phenotype, or its physical trait. And so this is going to show you the combination of these alleles and what the offspring might have. One important thing that you need to know is that a Punnett square doesn't show 4 kids. This is the probability of what one offspring will be. So there is a 75% chance that the offspring will be yellow. There is a 25% chance that the offspring will be green. Or if we're talking about it in terms of ratios, we would say 3 to 1. All right, so let's just remind ourselves what alleles are. So we have dominant alleles. Those are going to be represented by a capital letter. And we have recessive alleles. Those are going to be represented by a lowercase. All right, so some more terms. We have our parental generation, our first generation, and our second generation. So that parental generation is going to be designated by a P. F1 will be that first generation, and F2, and so on. You know, it'll keep going, F3, F4, and things like that. So the first thing that we're going to do, and you guys are only going to be required to do a monohybrid cross. Now there are dihybrids, there's trihybrids, and so you guys only have to do a monohybrid, which is pretty simple. And so mono is looking at one trait, okay? So we're going to look at the P generation, and we're going to determine the probability of these two parents being crossed. And how are we going to do that? We're going to do that through the use of a Punnett square. So the P generation, or a parent generation, so it is a female tall plant. It's going to be crossed with a male short plant. So the tall is its phenotype, and the female genotype is big T, and this is homozygous dominant, okay? So capital letters, again, represent the dominant allele. The male is short, that's the phenotype, and the genotype are 2 recessive alleles, or homozygous recessive. Sometimes you also hear the term pure. These are both pure because they have the same alleles. So we have our cross. We're going to cross this homozygous dominant with a homozygous recessive. So let's look at some steps to take. And you're going to do this for every Punnett square that you do. So the first thing you need to do is determine the alleles. Then you're going to set up the Punnett square. And then you're going to determine the ratios. So let's go ahead and do that. So the female is big T, or homozygous dominant. So what are her possible gametes, or alleles, that she's going to possibly give? Well, when these separate or segregate, she can give that first dominant allele or that second dominant allele to the offspring. So our possible alleles are big T or a big T. The male is homozygous recessive, and so what are the possible gametes? Well, they're either recessive T or a recessive T, or little t. Even though this isn't involved in this particular Punnett square, what would be the possible gametes or alleles for big T, little t? Well, we could either give a big T or a little t to the offspring. All right, so let's go ahead and set up our Punnett square. So female gametes are going to go on top, and males can go on the side. And to be quite honest, I don't really care, but if we want to have some continuity, we can put females on top and males on the side. So the letter on the top is going to drop down into the boxes below. You can kind of see the letters on the side are going to move to the right. So this right here is the Punnett square, and so you'll just draw a box and then 2 lines, so you get 4 small boxes. So we put our female here and our male right here. We're just going to drop those T's down, and these T's go across, and you've created your Punnett square. So now here's the most difficult part of doing the Punnett square, and that's determining the ratios. So we're going to do our phenotypes. So we're going to list all possible physical traits or phenotypes. In this case, it's going to be tall or short. So we're going to look here. So remember, the dominant always overshadows the recessive. So we know that's, and big T codes for tall and little t codes for short. And you'll always be given that information. So we have 1, 2, 3, 4 tall plants and 0 short plants. So sometimes you'll be asked questions like, what's the probability that you would have a short plant? And you would tell me 0%. Or what is the probability that we would have a tall plant? And you would tell me 100%. And so Punnett squares really show you the probability of something happening. So if it's not in your gene pool, if you don't have the genetic information to code for those traits, you are not going to express those genes. Now there are a few exceptions like some mutations, but that's very rare. Genotype are going to be the 2 alleles. So again, we want to list all possible genotypes. So homozygous dominant big T, heterozygous big T, little t, and homozygous recessive little t. Again, you're going to look at the boxes. In this case, we have all heterozygous genotypes. So 100% of the offspring are going to be a heterozygote. So now we're going to cross the F1s, which we saw on the last slide, to create our F2 generation. We're going to do the exact same process. So we're going to take this heterozygous, cross with another heterozygous plant. We're going to determine our gametes. In this case, the female can give a big T or a little t. The male is also going to give a big T or a little t. Notice they're separating, okay? So that's important to note, and we'll talk about what that is, what Gregor Mendel called this. So we'll set up our Punnett square. Again, we're going to put the female on top, male on the side. We're going to drop down those letters. All right, so let's determine our ratios. So our F2, remember we're still looking at tall and short. So we have 1, 2, 3 tall and 1 short. How do I know that? Well, because I know big T codes for tall, and if there's just one dominant allele, it will overshadow that recessive allele. Now we want to look at our genotypic ratio, so we're going to look at the alleles, so big T, little t, little t, little t, and just count. So we have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So 1, 2, 1. So from these findings and these mathematical data, he concluded a couple of different things. When I say he, I mean Mendel. So he came up with the law of segregation. So the law of segregation just means that only one allele is going to be given to the offspring, okay? So remember, you inherit half the information from your mom, half the information from your dad. So that's that law of segregation. So if a parent is heterozygous, okay, remember that only one particular allele is going to be given. If it's heterozygous, it's either going to be that dominant allele or that recessive allele. So again, you inherit only half of the information. The other law that Mendel came up with was something called the law of independent assortment. And we would see this more if we were going to do dihybrid crosses, which I'm not going to make you do. But what this means is that 2 genes are going to assort separately from each other. They do not influence each other. So if we talk about flies and eye color, and then we're going to talk about wings, okay? Wing shape does not influence eye color. So let's say if you have red eyes and normal wings, those 2 aren't inherited together, okay? They're inherited separately. And that's what independent assortment is. And it's another great way or reason why we have so much genetic diversity. So I like to think of your genes as a deck of cards. So if you are passing them on, you'll literally take your deck of cards, half it, and then you kind of shuffle it, okay? And those cards aren't attached to each other. They assort separately. And so that means that they don't influence each other when they're inherited or when they're passed down to their offspring. So kind of just shows you like, so the dominant versus the recessive, wild type eyes and body color and wing shape, they're all going to be assorted separately. We've been talking about it a little bit, but when we talk about probability, we should think about it in terms like coins, okay? So let's go ahead and skip. So if you toss a coin, what's the chance of it landing right side up? And so you would tell me probably 50% chance. So the probability is that 50% it would be heads or 50% tails. Okay, same thing if you have another coin. So 50-50. But what we have is something that's called the product rule. So the joint probability that both events occurs the product, and then you would put them together. So again, this is going to help you when you were doing bigger crosses, which we don't do. But let's say you have a mom who has a heterozygous genotype and a dad that has a heterozygous genotype. And what's the probability of the offspring having the recessive, homozygous recessive genotype? And so instead of doing a Punnett square, you'd actually do the product rule. So first you need to find the probability of having a recessive, excuse me, allele from mom and a recessive from dad. So you'd have 50% chance getting it from your mom and a 50% chance getting it from your mom, and you're going to actually multiply those together, and you're going to have a 25% chance, excuse me, I had to get a drink, of being homozygous recessive. So rather than doing a Punnett square, you would just use mathematics. All right, and this is kind of ending, and we'll talk about this more when we talk about DNA. But why aren't members of the same species identical? And it's really because of law of independent assortment, kind of that analogy I used of the deck of cards. So when you're given your genes, you're given a very unique combination of genes. Okay, so you might look similar to your mom or your dad, or you might look similar to your sibling, but you're not exactly the same. And that's because your genes assort separately from each other. So you're receiving a very unique combination of genes. The last one is mutations. And so mutations occur when DNA is changed. Now remember, DNA is the ultimate source. It's our coding, it's our information, our instruction manual. So if something happens in the instruction manual, then your traits could possibly change, okay? And so we'll talk more about mutations when we are talking about DNA, and it'll make more sense, but it's one of the reasons why species are not identical. So it's just showing you kind of that combination, just considering 100 traits and the mass amounts of unique combinations we could receive just from that. And mutations, it's just telling you that it's a change in the DNA and it's going to result in the appearance of a new allele. So we actually see this in populations where mutations create a new phenotype or a new physical trait. Now sometimes it can be harmful and result in death, but sometimes they're actually really beneficial. So you hear mutations and you might think bad, because when we think mutations in DNA can lead to things like cancer, but they can be beneficial as well. All right, and that is it. If you guys have any questions on genetics, let me know.